So it's my great pleasure to introduce Jiahui Guo. I try to prepare the practice, but it's not a new face here because Dr. Jiahui Guo is an assistant professor of psychology in the School of Behavioral Brain Sciences right here, also an external fellow at the Center for Vital Longevity. So we see her around, and now we're going to really hear in depth about what she's doing. Before joining UT Dallas, she completed a PhD in cognitive neuroscience at Dartmouth College with Dr. Bradley Luncheon, and her postdoctoral training also at Dartmouth with Dr. James Haxby and Ida Gorbini. She received her Bachelor's of Science in Psychology at Beijing Normal University, and her research, as we'll hear more about, focus on human high-level perception and cognition, combining research techniques such as naturalistic stimuli, neuroimaging, behavioral measurements, computational modeling, and artificial neural networks. With all this breadth, no surprise, her work appears in top-tier journals, including PNAS, eLife, Nature Methods even. So we're really fortunate to get a deep dive into what she's doing and how she's connected to everything that we do here. Thank you very much. Thank
you so much, Noel, for the invitation. Thank you so much for everybody here joining this, this talk. So for today, well, first I want to say my name, actually Jiahui is my first name and Guo is my last name, but always in the publication, I just split the two because it's easier for people to find my publication. So today I'm going to discuss the understanding human high-level perception using naturalistic. So actually, the world we live in is complex and dynamic. We need this high-level perception actually to help us recognize objects, to orient us in new places, to help us identify people, to read their expressions, understand their body languages, and also make social interactions. So this organ, the brain, which is about 3 pounds on average for human adults, protected by the skull, sitting at the top of the spinal cord, is the center for completing all those complex functions. However, after so many years of research, we are still not super clear how does the brain enable high-level perception in real life. Previous studies have already identified a few regions involved in this function. For example, FFA, here you might know, this is a region which involves in selectively responsive to phases. Those regions, most of those regions, they are in the back of the brain, in the ventral part of the brain, and also involves other categories. For example, there's also regions which are selectively responsive to bodies, to characters, to places, or to other objects. Those regions, they are not, they are not isolated. They are connected, and also extends to the more anterior part of the brain, even to the frontal lobe, to form we call that extended network. But after we know we have so many regions involved in human high-level perception, do we already understand how does the brain do this job? The answer is no. Why? First thing is most of the regions, they were identified using typical participants. And we know for a lot of those different abilities, we have this distribution. And the typical participants, they are at the center of this distribution, but we have people who are at the tails. We don't actually know whether for those neuropsychological populations, whether they are still doing the same thing, their brain acted similarly as in the typical participants. So we need to do research with these neuropsychological populations and to understand our brain from another perspective. And the second thing is, although we know those masks, we know those regions, but in real life, the individual brains, they have a lot of individual differences. We will call that idiosyncratic topographies. So for example, here the FFA, I just mentioned in those two brains, and we see they are wildly different in these two different individuals. So in real life or in real research, we need to also consider these individual differences. And the third thing is we know those regions, we know the locations. However, we don't know what kind of stuff is going on actually in those regions. In other words, what's the computations happening in those regions or the representations. So we'll need to do research or do studies to understand the representations of those things around us in those regions and in the network. So in my talk today, I'm gonna discuss those three different aspects using different kind of studies. The first part is I'm gonna discuss the research I did with neuropsychological population. I will focus on one special population, the people who have difficulty recognizing faces. We call them prosopagnostics. And the second part, I will introduce a method which can map individualized topography using naturalistic stimuli. This is a novel method, and I hope you find that interesting. And in the third part, I will try to connect the first one and the second one to apply this method to those neuropsychological population to see what we can get. And in the fourth part, I will try to understand the representations of those, in those regions of naturalistic. I will combine deep neural networks in this study to understand both the neural and the behavioral representations for naturalistic faces. Okay, let's focus on the first part. So faces, we know faces, they are very important. They are important resources in our daily life because we need to recognize the faces to identify people, to know their situation, to know how we can interact with them, and to complete our tasks. For our normal participants or typical participants, recognizing faces is very easy. We don't need a long time to do this task. However, it's going to be a super difficult task for this special group, prosopagnosia. There's two different types of prosopagnostics. The first one is we call them acquired prosopagnosia, or A. This group, as I show here, often have resections in their brain because of either a tumor or other accidents. They often performed normally before they got this brain lesion, but their ability was destroyed after they had this. And in another group, this is a group we call developmental prosopagnosia. Definition is in contrast. They don't have any lesions in their brain. They have face identity impairments in absence of brain damage or broad neurodevelopmental problems. There are two, very vivid examples of the personal experiences from those people. First one is I listened to an NPR episode recently and realized that other people didn't have to use shoes to recognize their brains. The second one is I once asked a friend to introduce me to the person with whom she was speaking and who turned out to be my friend. So they had a real difficulty even recognize their relatives, their children, and their family members. So of course, that's going to have substantial effects on quality of their life. And roughly 1 to 2% of the population were identified with this problem, which means 6 million Americans and 130 million people worldwide suffer from. In the past 20 or 30 years of research, the key question is try to understand the neural basis of this problem. However, previous studies suffer from a lot of disadvantages. First one is because this case where those people, they are, although like 1 to 2%, however, still it's difficult to collect a large number of participants in the study. So most of the studies they reported or documented in the literature had very small sample sizes, which means insufficient power to really understand the neural basis. And the second one is often they only studied the regions at the back of the brain or at the ventral part of the brain, which had only a limited coverage of the face system. And most of the studies only used static images, which limits their ecological validity of the study. And another one is often the method. If you want to localize the phase region, often you have to, after you scan the participants, you will have to draw a line to know, okay, this is the brain parts we want to extract to study. And you probably need a threshold, and we call that threshold-based method. Because the DP participants, they often have very weak activations in the phase regions, often using the same threshold for both the DP and control, you will lose biased estimation. So you will lose some of the participants in your study, which making it smaller sample size becomes. So in this study I did, we try to overcome all those challenges. First one is try to include as many as we can as possible. So we had, I want to say that's one of the largest sample size in the literature, although that's not a big sample size, but still that's pretty okay already. So we had 22 DPs and 27 controls in our study. And we didn't limit our analysis to the back of the brain. We tried to cover the entire phase network. And we used dynamic video clips in our study, which offered better ecological validity. And also we used the method we call variable window method, which didn't draw a hard line to select those voxels, which offer more objective and replicable method for the analysis. So the first question answered in this, we asked in this study is what's the, because we know the DPs have severe deficits in their behavioral performance. Considering those problems, do they still have a similar general organization of those phase network in DP as in control participants? So I plotted the peak coordinates of those category selected regions for both DP and control participants. And the darker, okay, this is not super dark, but the darker shades stands for the controls, and the lighter shades stand for DPs. And we can see for all of the regions involved in this high-level perception, the DP and the control, they had similar peak coordinates across the groups, which means although despite their severe behavioral problems, the two groups, they shared similar generalization for the entire the network. Then we dive down to into the details to see whether the selectivity in those regions are the same across the groups. Here I'm using FFA as example here, the red FFA as example here, and I define the phase selectivity as the difference between the percent signal change of phases and objects. And we plotted this phase activity in red FFA, and we found, okay, the DPs, they had a significant reduction of phase activity in red FFA. And the possibility is the phase activity reduction due to the weaker response to phases or a stronger response to objects. So we split the selectivity to the response to phases and objects, and we found the weaker phase activity was mainly driven by a reduction of response to phases rather than an increased response to objects. And the method I used was I selected the top 10% most phase active voxels in the mask, and we did analysis with other percentages, and we can see across all of the percentages here, this trend preserved. And this is not only true for red FFA, and I did analysis with all the other phase active regions in the network, and we found a consistent reduction of phase activity across all of the regions in the phase network in both the right and the left hemispheres. And we know besides those phase active regions, those high-level perception category selective regions, they also include the regions which are selectively responsive to other categories. So we examined the category selectivity in the polyselective regions and in the place selective regions. And we found the DPs, although their performance were mainly focused on phase perception, they had also consistent reduction of category selectivity in the polyselective regions and in the place selective regions, which suggests the phase problem might be the most noticeable problem in DPs, and their problem might also extend to other categories. And we call that the widespread reduction of categories. Let's summarize the results in the first part. So in this study, I found the DPs, they had normal general architecture for the high-level perception, despite their severe behavioral reduction or behavioral problems recognizing phases. In the second, we found the selectivity are reduced broadly in the phase active areas, and the reduction spread out to other categories like areas. And all the results suggested the face recognition deficits in DPs are only the most noticeable visual recognition deficit, which tells us, okay, in the future studies, the researchers might want to broaden their lens to look at the more broad, those visual recognition deficits in DPs and also in other. Okay, in this study, when we are talking about those category selective regions, we used a classic way to localize those regions. And this is something we call localizer. And this is the visual category localizer we used in this study. This is a classic block design study, which involved different categories of video clips. With this very classic way, if you want to localize other categories, you will have to scan, other localizers. For example, if you want to localize those, retina topic mapping, you will have to, ask the participants to do experiments like this. And we know from your own experience, this is not very interesting, right? So the localizers, although this is a very classic and very popular way to localize regions in your imaging study, it has different disadvantages. First one is often those localizers, they are impoverished and specific. If you need to localize different types of regions, you will often have to scan separate localizers for separate colors, which is not very efficient. You might need to spend an hour or even more on different tasks. For example, if you want to include like phase regions, you will need a localizer. If you need to localize those theory for mind regions, you will have to run a theory for mind study. If you need a retinal topic mapping, you will need to run localizer like this. You will need a pretty long time just to localize these regions. And most of the localizers, they are like this. I just had limited ecological validity. It's very difficult for the participants to maintain attention in the scanner. My own experience of this is I had tears on my face after I did this. Very a special experience. And of course, it's now friendly special groups, especially for the young kids or other patients. So we will need to consider whether there's other alternatives we can do to just overcome all these disadvantages. The alternative is one of the alternatives. So for the naturalistic stimuli, in contrast, it's often very rich and can activate multiple brain systems in parallel when you are just watching one movie. And movie is a pretty good, the replication of the our real life experience. So it offers better ecological validity compared to this published classical localizers. And it's more engaging in a scanner and very friendly to special groups your own experience probably will be if a baby is crying, you let the baby watch something is moving, the baby will stop crying. So similar experience, the kids and the patients, they are better, had a much better compliance in the scanner than just use this static. So but the question is, if we use naturalistic study, how can we get those regions we need from this rich and complex the visual inputs, visual and audio inputs? So in the second part, I want to discuss a novel method which can help you to solve this problem. So our question here, let's rephrase this. We want to predict the topography of a participant using just the movie data of this participant. To do this, we will need another data set. In this data set, this is an independent group of participants. They need to have the movie here also run the same movie, and also they will need to have only together. By having this data set, when you have a new participant, you don't have to participant to run the same local ladder. You just need to ask this participant to watch the movie, and you can get the individualized topography using this method. So how can we do this? This is a method we use called response alignment. So I will tell you how to do this in detail. So let's put the data here. So this is the participants, the training participants here, and I just put it on the left side in the last slide. And this is the participant we want to predict. We want to predict this participant's topography here. And if we treat each vertex as one dimension here, the response to the movie we can plot here as a vector in the high dimensional space. So for each of the participants, we have a response in the high dimensional space here. And similarly for this, the participant we are going to predict. And then the next step is we're going to rotate this high dimensional space for every training participant to match the vector in the high dimensional space in this testing participant. The method we used is basically based on the Procrustes method. And then we can get the transformation matrices. We call that rotations for every participant. And then the next step is we can apply the individualized transformation matrices to the local ladder data of the training participant here. So after this step, the data of the local ladder data for each of the training participants will be in the space of this testing. Then we can do a classic general linear model to see, okay, this is the map where the topography we predicted after we do this. If we don't do any of these rotations or transformation matrices, or doesn't do anything and just do a general linear model, this is the most classic way to analyze the data. We can get this we call surface alignment prediction. So I will use the face selective topography as the example here. So this is the topography we got from one example participant, and I zoomed in on the ventral temporal cortex for you to show you the results more clearly. First, let's see. This is the results we got from this participant using the most classic their own local ladder data. And if we don't do this, and if we just predict the map using other participants, what we can get. This is the map we got using the hyperalignment method I just mentioned. You can see the prediction on the predicted map looks very similar to the map we got from their own local ladder, which not only recovered the general shape, but also like the little creature, those little curvatures, and this more fine-tuned or fine-coarse activations here. If we don't do this alignment and just do this surface alignment, and this is the map we get, and we can see the map is blurry and doesn't have any participant-specific information here. And then this is the result from another party. You can see they basically followed the similar pattern here. If we correlate, we need to quantitatively test whether this is a good estimation. So we correlated the predicted data, the topography to their own localizer between the hyperlined, the map we got from hyperlines and the maps we got using the surface element. And now let's out here. So we see the correlation we got between the maps we got using hyperlines and their own localizer at a much higher correlation or much better performance than between the surface alignment map and their own localizer map, which means the hyperlines outperformed or significantly outperformed the surface alignment here. And we plot the noise ceiling from our data, and we can see the prediction is very close to the noise ceiling too, which means this is a really good estimation. And we used another data set to validate these results, and we found a similar pattern here. And we plot the individual results in this plot. The blue dots are the one data set, and the orange dots are from another data set. And the x-axis, the performance using surface alignment, and the y-axis is the result from the hyperalignment using our method. And we can see for every participant, the results we got from our method outperformed the traditional surface.
Ask for clarification. What you're demonstrating is the benefit of the hyperalignment procedures towards functional localization, right? You still have to do the same process of labeling the segments of the movie and then contrasting, or are you not doing that at all?
No, you just take the movie as the entire time theory. You don't have to do anything. You just use the method to rotate you will treat their responses in the high-dimensional space and rotate their responses. And you don't have to really label the movie and do anything else.
So then how do you identify the appropriate map to contrast with your localizer?
That's the good question. So you basically understand that, like, you build the connection between like one participant and another participant by connecting their responses in their movies, using the movies. And then you will apply these individual differences to their localizer data of the training participant. So by doing this, you can get the predicted map using this method, just by remembering or getting the connections between the movies.
So just to follow up for clarity, so this is the faces versus other.
Yeah.
That is, you're looking for the similarity in the training. It's for faces versus other localizer conditions.
No,
How
you can use
some faces versus others.
Well, so I can go back. So here, for this average training participant, you have the movie data and the localizer data. This is your must have. You must have the localizer data in this training participants. And for this testing participants, you can only have their movie data. But this is where demonstrating this method, we are using their localizer data to validate whether the prediction is great. But in the application scenario, you don't need to have the localizer data for this.
So how do you use the face localizer in the training?
So here, you first get the transformation matrices between by rotate their responses of movies in the high-dimensional space. And then you apply those transformation matrices to the localizer data of the training. And then you will have the time series of their training participants in the testing participants space.
By apply, do you mean make it select faces?
No, you get these rotations where this is a matrix for each participant. And then they have their, their localizer, their runs. And then you just apply this rotation to their localizer runs. And after this, their localizer run will be in this testing participant space. And then you just do the normal, the GLM, and then you average across all the training participants to estimate the map in this testing. Clear?
I'm still not sure where you select faces?
Select
How do you
faces
make
in the
it select
training,
yeah,
in the
faces?
training set. It's in the training.
The training set does have plus one of the faces and
the local ladder.
Anything else?
Yeah,
Or
the
something
loc-
else?
Yeah, so until this step, after you rotate these local ladders, there are still the I want to say time theories from the scanner. It's just the sequence you got from the scanner. And then you do the GRM, it's basically doing the contrast between the faces and other categories. You can also, if you want to do this, you can also do faces versus objects, faces versus houses to get the regions you need. You can do whatever you need. But this method just prepares you to do anything else. Okay, so the method I just introduced requires every participant to watch the same movie. However, in the real scenarios, it's very difficult for the participants from different part of the world watch the same movie because the data could be collected from different institutes, and the participants they can speak different languages, and they'll often need to tailor your movie in order for the participants to understand what's going on in the movie. And also the data set can be collected from different scanners, and even using the same scanner could be using very different parameters. So you don't have the time theories across all the participants. And we need a method actually to generalize across all these kind of differences. So in this method, in this study, I want to introduce a more like optimized method which can overcome all the diverse situations. Just to rephrase our question here, so we want to also predict the topography of the test participants. The test participants can only just have the time theories from watching a movie. And for the training participants, they need to have the local ladder and also a movie data. The movie can be a totally different movie. The language can be different, the scanner can be different, anything else can be different, as long as it's a task. So how can we do this? This time we will use a method we call connectivity method. If you still remember this map or like this figure, this time the difference is we will need to calculate the connectivity profiles before we do the rotations. So here, there's an example here. We will need to like how we build this connectivity profile. That's we calculate the correlations between each vertex in a region to the other vertices across the entire frame, and or you call that target seeds. So here, this shows the connectivity profile between every vertex in this region to the other targets in the brain. And after we have this connectivity profile, we can put their connectivity profile in this high-dimensional space. This is in the high-dimensional space already. And then we will need to also rotate their connectivity profile between each pair of the participants in the training and this test data set or test participants to derive their transformation matrices for each of the training participants. And still similarly, we will apply these transformation matrices or rotate these transformation matrices or apply these transformation matrices to the local ladder data individually of this training data set or training participants. And then still after this step, the local ladder data will be in the same high-dimensional space as in the test participants data set. And then you can do or in this particular study, I did several iterations in order to improve the performance, but this is more just the details. If you are interested, we can discuss after that. And then you can also do the general linear model, do the contrast to get the topography of whatever category you like. And we call the map after this step is the hyperelement or connectivity hyperelement predicted map for CHA. And similarly, if you don't do anything, you just collect your local ladder data and do the general linear model, do whatever. This is a classic way. You will get this surface value predicted map. And whether first we want to see whether the new method can perform similarly as we had in the the response hyperelement we introduced earlier. So here, this is a map from one particular participant using their own local ladder. And this is the map we get predicted from the other participants. And this, like the training and testing participants in this scenario, they watched the same movie that's in the same data set. So we can see if we use the new method we introduced here, the map we predicted were very similar to the map we got using their own classical ladder. Similarly, if we just use the surface element and the map we predicted were just also very blurry and without any specific personal or individualized information. Then the next interesting thing is whether we can do the similar prediction using another different movie. So whether we can do this cross-movie prediction. So here we see if we use another movie, this is a map we predicted by using a totally different movie. The language are different, as in the one movie was collected in the US and the other data that was collected in Germany, and the movie are totally different, the lengths are different, everything's different. So here we can see the map we predicted were also very similar to the map we got in their local ladder, and also very similar to the map we got using the same movie. And we plotted the individual data here, and the x-axis is the between movie prediction, and the y-axis is the within movie prediction. And we see basically they are the same. And this is not only true for one category basis. This is also true for the other categories. If you need to get the body selective regions, place selective regions, or object selective regions, they're the same. And then we plot the stats results here. And we can see no matter that's the within movie or cross movie prediction, they are both outperformed the surface element predicted results, and also very close to the noise ceiling. And this is true for all the 4 categories we can do in the. And we also introduced other different data sets and similar results of data sets. So in all of the results suggested the individualized topography can be predicted with high fidelity across diverse situations. So as the summarize here, part 2, we introduced a method which can do the individualized topography estimation with high fidelity from natural space. And then with an optimized station method, we found individualized topographies can be accurately predicted across different diverse situations. And then this method provided us a tool for integrating those high-level cognitive functions across datasets from laboratories worldwide. Imagine you have your in-lab collected data, and you have another dataset, public dataset, and you need to actually derive those regions using, in those participants who didn't collect any of the local other data you need. You can use our method to bridge the gap and get the regions you need from those public data. And in the third part, I want to combine the first part of research and the second part of research to see how we can apply this method in the neuropsychological applications. I actually included 2 studies here, but I don't think I can have to finish both. Let's focus on these DP participants first. Okay, so the question asked is, can we predict the individualized topography in the DP participants? So rephrase the question is, we want to predict the topography in the DP participants using a different task. The participant doesn't have to, scan the local ladder data. They scan the local ladder, they just need to have a task. And the training participants can be either the other DP or the control participant. This is more important thing. They need to have both the local ladder data and another independent task. Let me do this. Okay, so this work was done by my lab manager, Ian. And in here, I'm gonna also show you the results from the faces versus others, which means that's the face selective topography here. This is the results from one DP participant. And it looks, from the DP participants' local letter data, it looks like this. And here I'm showing you the map we got from the estimation using other participants' data. So here, this is the results we got using the same group of participants. They are all DP participants, but different individuals. We are using the other DP participants to predict the left-out participants' data. So we used both the response hyperelement and the connectivity hyperelement to do this. And we found the map we recovered were very similar to the data from their own local letter. And similarly, the surface elements were anatomical elements, but then do a good job here. And most importantly, can we just use the control participants to predict the topography in the atypical group? Here, if we use the control participants, this is the map we recovered here, and we found they did a pretty good job actually, using a totally different group to recover this atypical group participants topography. And for, on the other hand, how can we do this in another direction? So this is another participant in the control participants group. And then we did the estimation using the other control group. This is basically a replication of the data or of the results I just showed you in the part 2. Here, so not surprisingly, the maps we recovered were very similar using either RHA or CHA of the maps we got using their own local letter. But can we do the reverse direction? If we use the other DP participants to predict the map of this control participant, can we get a similar prediction? The answer is yes. The maps we recovered were also very similar to the map we got using the local letter, their own local letter data. Although the training participants are from a atypical group. And here I showed you the stats, and we plot the bars here, and that's the results from the RHA, CHA, and also the anatomical alignment. And we can see for all of the categories, if we use the other DP participants to predict the left out DP, the results were very good. So both RHA, CHA outperformed the anatomical alignment, and they also outperformed the noise setting here. If we use the control participants to predict the DP, the results are similar. And for all of these cross-group predictions, we actually found a very similar prediction performance here, which means our method can not only cross different, different tasks, different movies, but also can cross different groups here. And if we plot these individual data points here, and different colors, they stand for different categories. And we can see for every individual, in every these cross-group predictions, the RHA or CHA, they both outperformed the anatomical element in a steady manner. And you'll probably want to see whether the predicted map also performed or acted similarly as if we just derived from their own local letter. So here, Ian did a replication of the results I showed you in the first part, the DP participants, the reductions across all of the phase network. Here, we just used the predicted topography here, and we found this similar consistent reduction of phase activity across all the regions in the phase network. And the results were also mainly driven by a weaker response to phases rather than a high response to objects. And we used another data set because the first data set we used doesn't have the naturalistic data set, just doesn't have a movie. It had another controlled classic task. We want to see whether our method can be applied also in the naturalistic situation using just a movie. So we used another data set, in which the DP and control participants, they watched this movie, The Game of Thrones. And in this data set, but this data set only contained a very crappy, just one run, a short, noisy local letter. But still, with this very noisy data, we found that our method consistently outperformed traditional anatomical alignment. Also for every individual, this is a pretty big data set. So for every individual here, you can see this very steady and very consistent good performance in both RHA and CHA. And see here, the correlation is very low. Why? Why this is the scenario? And if we plot the results, you will find out, okay, because this local ladder run is very noisy. Here, this very noisy data here. But with this static, short, noisy data, this local ladder itself can only basically localize this static bilateral regions here at A. With this noisy data, we can see the maps we predicted were actually less noisier than the original data. So here you can see this those frontal regions and the motor cortexes, they have much lower noise than this one. But for this most important regions, this local ladder itself can do, our prediction is actually very accurate. For even either using DP to predict DP or either using control to predict DP. The low correlation was mainly driven by the other noisy boxes here in the data rather than our predicted map. Similarly, this is the result from control participants in which and the other estimated map from using the other control participants or using the DP. And we found even with this very noisy data, we can still get the regions we need from using our methods. So in the third part, I demonstrated the individualized topography can be estimated with high fidelity in this DP participants. We found also excellent estimation can be achieved by using either a task data, traditional classic task data, or using the naturalistic movie data with CHA. And this bridge the gap between the current method and the future applications, and we can apply this method in not only just the DP groups but also the other atypical populations. I have another result from application to the blind and the deaf participants, but I don't have time to go over those. So let's quickly into the first part. The first part, let's see how much we can do. In the first part, I want to introduce the representations. So I want to just combine also the artificial networks and also the fMRI and also the behavioral testing to see how we can bridge the three. So we see those real faces every day. These real faces, they talk to us through this changeable aspect. For example, the expression, their eye gaze, and their head orientations. This is very different from the experience we see just a static photo. However, in the previous studies, most of the studies trying to understand face perception, they use static images, which couldn't tell us how our face perception are doing in the real life. So in this study, in order to answer how we represent the naturalistic faces, we build a high-dimensional face space using a large number of naturalistic face clips, and we call that hyperface. And here are two examples from this hyperface. This dataset, which includes 707 naturalistic video clips, unfamiliar faces, and each video clip is 4 seconds long. And then the video clips, they vary badly in identity, age, ethnicity, facility, and other dimensions. And it's one of the largest naturalistic face village set in the literature. So we use the multimodal approach to answer or to bridge the DN, the brain, and the behavior. So with the DN, we use a face-trained face DN here, not A, but Phil, but there are phase 2 DN, and we put our video clips into this network and get embedding vectors from every layer of this network. And then with these embedding vectors, we can build a DCN representational dissimilarity matrix. Here, RDM stands for this dissimilarity matrix. So here, every cell in this matrix stands for the dissimilarity between each pair of these video clips. Then we had another dimension. That's our behavioral performance. So in this behavioral task, we asked our participants so first we put the faces, the video clips around this circle and ask the participants to drag the faces based on the facial appearance or based on the similarity of the face identity here. And this is the result from one particular participant. So here you will find, very interestingly, the participants, they actually group the faces based on first dimension, the gender of the faces. They put the, the male faces on one side, the female faces on the other side. And also we see that, they also put the faces based on the age. The young faces are here, and the other along this dimension, the older faces are there, the young faces, older faces. And also we found that, okay, here, the faces with darker shades, they are more to the inner part of the circle, and faces with lighter shades, they are more to the outer side of the circle. So the participants, they are, interestingly, this is only one participant, but with eyeballing nearly every participant, they perform this task using a very similar manner of this behavior. And then after we have the results, we can use their physical coordinates to build also a physical, this behavioral representational dissimilarity matrix. And the third one is the neuroimaging part. We ask the participants to watch those 707 faces in the scanner, and then we use the search analysis method, and we get their the pattern of their face responses from every search height. The search height size was 10 millimeters in this analysis. And with the pattern of the responses, we can build a human neural representational dissimilarity matrix here. So then we have all the three different types of representational dissimilarity matrix, and then we can do something. We asked the two key questions here. The first one is, do face DCNs, the human face clustering behavior, and the brain, do they share similar internal representations? Which means that we will test the correlations between the three different RDMs. And the second one is, if we find any similarities or dissimilarities, what's the underlying dependence driving those similarities or dissimilarities? So let's go from the first pair, the DCN and the behavioral RDM. So we did this for the average layer of the deep net here, and we found the correlation were pretty low in the beginning and gradually going up until it reached the top and then going down. And if we plot the noise ceiling from our behavioral data set, and we found actually the DN, they explained a lot of, or nearly 90 or even 80 or 90% of the explainable variance or meaningful variance in the behavioral data set. This is not only true for just one deep net. We involved another two phase trained deep nets, and then we found they had very similar, the performance or similar pattern here. In order to understand why the DNA and the behavioral representations, they share this strong correlation, we involved another behavioral study here. So we asked another independent group of participants to label those faces along these several dimensions. First one is the perceived gender, whether the face is a male face or female face, what's the age range of the face, and what's the ethnicity of the face, what's the gender expression, and what's the overall head orientation. And then after we got those labels, we created an index. And this index is the difference in the base for the spatial distance of the between and within feature groups. What's this mean? So here, every dot here stands for one particular video clip. And we plot this in this we just do this in this DS plot in this two-dimensional space. And the orange dots, they are female faces, and the blue dots are the male faces. So if we have a high index here, the larger this index is, the cleaner this division is between the two clusters. Okay, so for this arrangement task with our eyeballing, they basically cluster the female faces and the male faces together. So they have a very good, very big this index. But for the arc face, the deep net, the best layer is not, had a pretty good clear cut. But the final layer here, there's cut, but pretty mixed together. So we plot this index here, which shows whether there's a clear division between different clusters. And we found that for this behavioral task, this task is mainly driven by the gender, age, and ethnicity, which is very consistent with eyeballing data, right? If you remember those clusters. And we also plotted this index for every layer of the 3 deep nets we used. And we found this index were just low in the beginning and going up and then going down. If you remember the pattern there, the correlation between the deep net and our behavior, this shape is very similar. Okay, and together considering this, basically why the behavior, our human behavior and the deep nets, they share those correlations. That's because they're basically mainly driven by those, we call it categorical major informations there. So the deep nets also captured those the gender, age, whatever this categorical information as in our human business. Okay, so the DNN and the behavioral representations, they capture them. Then let's go to the DNN and the brain. So we did this for every small part, the search size for the brain, and then you can go over the entire brain to have the coverage. And if you do a correction, this is the regions we got after we do the correction, which means the highly correlated regions, they are basically the regions you can imagine. They are the visual regions, the face selective regions. And then we calculated their correlations for the average correlation in the face selective and the non-face selective regions here. And we found for every layer, the correlation in the face selective regions are higher than the non-face selective regions. This is also not only true for one deep net, and so for the other deep nets, which means the deep nets actually capture the meaningful variance in our brain. However, if you see this correlation strength, it's very low. It's smaller than 0.1, which means less than 1% of the it's not only less than 1% of the variance work can be explained by this correlation. Does this mean we had crappy data? Why this is the correlation is so low? So we actually tested the noise ceiling of our data set, and we found those regions we are mostly interested in had a very high noise ceiling, which means we had pretty reliable data, just the correlation itself. Okay, and then let's go to the behavioral and the brain. You will see probably the behavior can explain the brain variance, but the case is not there. So we did a similar analysis for every search ad here, and however, we found the correlation between the behavioral RDM and the neural RDM, were meaningful. Yes, meaningful. So the face selective regions had a higher correlation than the non-face selective regions. Yes. However, this correlation strength is very low too, which means the behavioral, just the one type of behavior, can only also explain a small part of the total variance of the rich information going on in the brain. And then we also plotted this index here for the RIs. This is the results using the right FFA, and this is the perceived gender here. This is the perceived gender here, and I plotted this index for every RI in the face network, and we found the highest the highest values are coming from this right ventral temporal cortex, which is consistent with our prior knowledge that the ventral temporal cortex, the face regions, they are better at representing those static based information like the identity or gender or ethnicity. Okay, let's go here, and then we plotted the values for all the other categories here and in all of the space selective regions, and we found this is very meaningful too. Like for age, it's also mainly captured by also the right hemisphere ventral temporal cortex, and for the dynamic information like expression, it's better captured by the right hemisphere dorsal network, which is also consistent with our prior knowledge. Which means all the information represented by the brain is meaningful. However, those information also only captured a small amount of information in the brain. Because if you remember, like the index there, it was very big for the behavioral task, but very small for all of the. So let's do a summary here. So we first found the representations of the FSDN and the behavioral clustering task. They captured a similar major trace features. And then we found the FSDN and the behavioral clustering task modeled only a portion of the variance in the neural representations of naturalistic physics. And the third, we found the neural representations of naturalistic physics, which contain the information beyond just the major categorical information we showed will include. So in the future, we will need to further understand the other part of the information presented in the brain, for example, like memory, attention, and other dimensions. And we had another study going on in the lab, and one of the students, they are using the test to model to see whether the fitness can model the social traits judgments of them. So if you are going to sense 2 weeks, 1 week later, still by our question. So let's go back to the major topic here. So the high-level real life. In this talk, I discussed this high-level perception from the 4 different perspectives. First one is neuropsychological research. I discussed the base activity and distribution for the general architecture in deep evidence. And then I introduced the method trying to map the visualized topography using naturalistic stimuli. And in the third section, I combined the 2 different parts to see whether we can apply this method to the atypical groups. And in the first study or first section, I try to discuss the representations in those physical regions using naturalistic. Then I want to give my thanks to all of the people who are very important in my journey, and also thanks to all of the lovely members. Thank you so much for sitting here for such a long talk, and I know. Thank you so much. Ready for
Any quick questions?
I have one, which is what struck me from the analysis of your very beautiful hyperalignment is that unless you're really interested in very fine-grained topography of, for example, faces in people, there's no need to do it because the surface map is pretty good. It's just a defocused version of the individual topography. But if I'm interested, for example, in picking RORs, or I'm interested just in getting a general blurred version where most people have their face responses, I don't need to do it. Well, surface mapping is really good. It's just a defocused version of the individual maps.
Yeah, I agree. I think this really depends on like the purpose.
Yes,
of the study.
it's the question that matters.
Yes, yes, but most of the time, you really need to get this visualized version. Sometimes this is very important for some of the studies. This is the recommended. But if you only just want to get a general application and a general application, I think building the surface map.
It's been pretty good.
Yeah, it's so.
Just a little bit of follow-up. I guess I'm still trying to think about the application and what you put as a need for the localizer, those training subjects. Just a basic question, though. If you just took the movie data and just did a contrast of face segments versus non-face segments, can't you just get face localizer? And if that's true, you'd be able to just split the movie data in half, use the first half for a localizer and the second half for hyperalignment training.
Good question. Actually, one of my colleagues before she did an analysis trying to contrast, but this is just a very ideal. If you want to do this, you first have to do this applications.
Sure.
So just a pain in the butt. And the second thing is for a lot of movies, for example, if you want to do this contrast, you want to have a balanced tempo basis. But this is often not true for most of the
But you could
movies.
select the frames in a balanced way.
But don't forget, our hemodynamic response is very slow. is also now very accurate. Just the movies are changing, frames are changing fast.
Yeah.
Our brain response is very slow, which makes the later analysis not. It's okay, but not as good as doing this. That's first thing. The second thing is this method can be generalized to other types of factors, not only face, but also if you want to do a calculation, you need to get calculation regions and therefore mind regions, or get, for example, red topic mapping. You can all just using this method, but you will have to rely on the content of the movie if you have to do this contrast. That's
Yeah, I mean, for me, that's actually a feature, right? Because you open yourself up to a broader set of localizers embedded in whatever the content of the movie is.
And the third thing is you don't even have to have content. The classic task can also be applied there, and also resting data sets. You can also just scan a resting and get those regions. You don't have to rely on any of the content. And also you don't have to scan very long. Our analysis movie is very long in most of the scenario, but the Game of Thrones, that one is only 13 minutes and very noisy. Still we can do.
If I can push you more as a kind of follow up of those, first of all, great thought. So for faces, we know where we're looking for any kind of visual object category, visual categories, we know where to look. How good of a localizer what does it really translate to beyond those very well specified categories? And like even within that, it might break if you have, I don't know, a table with legs. What do you do? Like you have to kind of buy into those modularity, and then you find the modularity. But is it really that our brain works in this very modular way? And if it's not, then where would still be the so like faces? Just an example of a localizer you think it work for that is not visual, selective, specific category.
Yeah, and this method can be generalized to not only visual categories, but also like more abstract regions. For example, the one always said multiple times today, the zero to mind regions, and also the calculation for other whatever regions you can imagine, because this doesn't really rely on any of the real content of the movie. It captures the general individual differences. So we actually applied the individual differences we captured using the general results of the movie to another type. As long as you have this in your own data set, you can recover the other percent regions, the data that person doesn't have.
Any questions online?
Yes, we do have one question online. It says, how do artificial networks show how to improve distortion of real world scenarios in neurodegenerative disorientation typical of Alzheimer's disease patients?
Can you say that? Can you read the question again?
How do artificial networks show how to improve distortion of real world scenarios in neurodegenerative disorientation typical of Alzheimer's disease patients?
I don't think I got this question very clearly, but I think if I rephrase this, I can say, I think the question is asking whether we can use deep nets to understand Alzheimer's disease. If I have to try to simplify this question. So if I want to do this, is while the deep nets part, I didn't include any of the atypical groups in the analysis. And I also don't think the deep nets, my current deep nets is good enough to capture the atypical groups either. So we are still trying to understand first whether the deep nets can capture any of the typical groups, the brain or their behaviors. And then after we do this, we can then to see whether the deep nets can be applied to understand the data from atypical group. But still, we're on the first stage, and this is a future topic.